Hi, good afternoon, everyone. My name is Felicia Brundick. I am the Outreach and Education Coordinator for the Missouri Prairie Foundation. Thank you for attending today's webinar, The Role of Mycorrhizal Fungi in Prairie Remnants and Plantings, with Dr. Alice Tipton. Mycorrhizal fungi play an essential role in structuring plant communities, and this is particularly true for prairies. In this talk, Dr. Tipton will explain how mycorrhizal fungal inoculation can act as a tool in prairie reconstruction. We are excited to bring you a lineup of masterclasses and webinars over the next few months based on the feedback from our summer webinars that we've had. Dr. Alice Tipton of Columbia, Missouri, received her PhD from the University of Missouri in 2016 and conducted postdoctoral research at the University of Kansas, working on projects testing how mycorrhizal fungal inoculation can act as a tool in prairie reconstruction. She is currently a professor of biology at St. Louis University in St. Louis, Missouri. After the presentation, Alice will have time for a few questions, so please type your questions in the chat feature below or the Q&A section, and we will address as many as we can. Please also make sure to visit our website at moprairie.org to see all of the great upcoming webinars and masterclasses and other events, such as our prairie picnics coming up this Saturday, October 31st at 5 different MPF prairies, as well as the 2020 Grow Native Professional Member Conference, which is open to all this year virtually, and it features keynote presenter Dr. Sarah Bayer, who will speak about the carbon storage capacity of native plants. It's going to be really exciting. All right, thank you very much for listening to me talk, and now here is Alice.
Thank you very much. And can you hear me? Great. Okay. Thank you all for being here. This is one of my favorite things to do. I actually had a meeting with a student earlier who was interviewing me about kind of career path, and they had to do that for a class, and she asked what my favorite parts of my job were, and talking to people like you, I told the student, well, I get to talk to a group of people today. It's one of my favorite things to do, because it's like talking to a classroom about something that you find really exciting, and most of the people in the classroom also find it just as exciting as you do. So I'm really, really happy to be here and hope that we can all spend some time learning together. So I'll be talking about the role of mycorrhizal fungi specifically, but microbes in general in prairie remnants and plantings. And I'll be looking to the side during this. That's because my big screen is over here. So if you see me doing that, that's what I'm doing. Okay, so as many of you in here probably know, and we can kind of categorize prairies in a couple different ways. I'm not going to spend time talking about why prairies are important or what a prairie specifically is, because I might assume that most people in this webinar probably have an idea about that. But I do want to review some terms that I'm going to be using and some that I might be using interchangeably, so we can think about the different kinds of prairies. And mostly what I'm going to be talking about is the difference between remnant prairies and reconstructed prairies. So when I think about remnant prairies, and I think about remnant prairies in my research, I'm thinking about sites that have never had a large enough disturbance where prairie plants had to be reintroduced through seeding. And so these are what I call remnant prairies. Now, in the state of Missouri, a lot of the prairies that I worked in, and some which are managed by the Missouri Prairie Foundation, are often sites that maybe have had some type of post-European settlement agricultural disturbance, but those fields might have been used as a hayfield and never been plowed or never been heavily grazed. Or they might be sites that never went through any post-European settlement disturbance. And so those remnant diverse prairies remain without any seed additions. But then we have sites, as many of you know, prairies are a great place to conduct agriculture, both in terms of grazing cattle or for planting monocultures like corn and soybean rotations here in the Midwest. And so sometimes when these areas become vacant, or what we call fallow fields, and we have an opportunity to put prairie plants back into the place, into the site, I'm going to refer to those as reconstructions, and that would be like a planting, some place where seed addition or plant additions has to occur in order for it to get that native plant community back. Now, this process can be a really challenging process, taking these sites that have had a huge post-European agricultural disturbance and trying to restore these native prairie plants. But lucky for us, we work in a system there, the ecosystem restoration is common throughout the world, trying to take these degraded places and put them back to their native communities. But in prairies, we're really lucky. And I have to always remember, one of my colleagues says, that we get prairie plants back. It's really exciting. This is a prairie in some regard. These are native prairie plants. But this reconstruction never quite looks like those remnant prairies, those sites that have never been plowed or heavily grazed. And that's because, one, the diversity seems to stay lower, but also the structure of this plant community is different. So for example, I like to use these cartoons that my colleague Elizabeth Middleton made. If we think about a remnant prairie, and if we're looking at a plot in a remnant prairie, what the composition of the plant community might be, we'll have some dominant species, for example, big blue stem or little blue stem, that seems to be pretty common. And then we'll have these species that we'll call mid-successional species, something like brown-eyed Susan, these other forbs. But then most of the diversity in a remnant prairie comes from these plants that some people call late successional. So there's species that you really can only find in these remnant prairies or more specialized to prairies, and they're really what makes up the diversity in these places. In reconstructed sites, even though most of the seed is added to these locations, some of those late successional plants either come back in really small amounts or maybe not at all. So for example, in these reconstructions, maybe the dominant plant is the same. You have a lot of big blue stem or a lot of little blue stem, but instead the diversity is lower, and it's mostly coming from those mid-successional plants, with a very few of the ones I've circled in purple, what we would consider late successional members of the community. And sometimes this effect persists for decades after a reconstruction. So why? Why are reconstruction or restored prairies different? Well, there's lots of things that we could be raised in our area for thousands of years. Fire was a really common tool used by Native Americans. These prairies took a long time to establish, so maybe decades just isn't enough time. It could be a seed dispersal problem. There's a researcher at the University of Missouri, Lauren Sullivan, who works on prairie seed establishment and dispersal. Maybe it's just not enough of certain plant seeds that are getting to these locations. It could be invasion by non-native species. Sometimes it's very difficult to keep the non-native, non-prairie plants from taking over in these places. It could be the soil nutrient levels after agriculture. Oftentimes, for example, things like phosphorus or nitrogen levels can be really high. Maybe it's not a good soil environment for these prairie plants. But then the last one, which I'm mostly focused on, is the soil microbial community. So are the microbes that these plants need, are those present in these reconstructions? So let's think about what I mean by soil microbial community for a second. And I'm going to start by reminding everyone something that's been in the news a lot lately is this idea of the human microbiome. So for example, we're learning more and more about all of these other types of microbes and bacteria that live in our body, in different parts of our body, and how important they are for our function. And in fact, there are more microbial cells in the human body than there are of our own cells. We know that we have this human microbiome that's really important for our health. Well, we can think about plants, and especially prairie plants, in the same way. So plants also have a microbiome. Some of the microbiome, you might be thinking of the above ground part, but I think about the soil microbiome community or the soil microbial community, all the microbes that each of these plants and this really diverse remnant prairie are interacting with below ground. Now, this community could include things like pathogens that maybe decrease the growth or the survival of certain plant species like nematodes, specific type of bacteria, fungal pathogens, but it can also include different microbes that are really important for helping plants grow. And these would be usually, but not always, considered mutualist, where both the plant and the microbe is benefiting. Examples of that you might have heard about would be something like rhizobia, that's present on legumes and helps fix nitrogen. And then the group that I study, which is mycorrhizal fungi. But I want to make sure I say here that though I'm going to be focusing on mycorrhizal fungi, when we're talking about the diversity in a prairie, both the good and the bad, both the pathogens and the mutualists are really important for keeping this diversity here. But I'm going to focus mostly on the group that I study, which is mycorrhizal fungi. The other one I want to keep in mind is that there's a lot of them. If we think about the soil community, just like we think about the human microbiome, there's tons of different species, some of which we don't even know what a species is for some of these microbes. This is an early paper in 1998, and even back then we knew that there were tons of bacterial cells per gram of soil in different ecosystems. And I have the ones here for temperate grasslands. That's a big number. That's how many cells there were in 1 gram of soil. And then bacteria, but bacteria aren't the only ones here. We have, it's kind of showing the soil community in general, including both microbes and larger macroorganisms that we can see, but rhizobia, other types of fungal pathogens, mycorrhizal fungi, other types of fungi called oomycetes, all of those are important for the soil microbial community. But I focus on a specific group called mycorrhizal fungi. And mycorrhizal fungi are a group of fungi that live in and on plant root systems. And these fungi help the plant take up things like nutrients and maybe sometimes even water that the plant can't get on its own in exchange for some of the plant's sugar resources that it makes during photosynthesis. Now, there are really different types of mycorrhizal fungi. This is a picture of a type of mycorrhizal fungi called ectomycorrhizal fungi, which isn't common in prairies, but I like it because you can actually see the hyphae of the fungus. So this is a picture of a pine seedling interacting with ectomycorrhizal fungi. And just so you know, about 90% or so of all land plants on Earth interact with some type of mycorrhizal fungus. So this is a picture of all of the hyphae. This is the roots of the pine seedling. And then all of this is the hyphae of the mycorrhizal fungi. So the mycorrhizal fungi not only is helping the plant get nutrients maybe in forms that the plant can't get on its own, but it's really extending the surface area over which this plant is interacting with the soil. Now, the main group that I study and that is most common in prairies is arbuscular mycorrhizal fungi, or I'll call it AMF. And AMF gets its name because it forms a special structure inside of the root. So this is a much more microscopic group of mycorrhizal fungi than ectomycorrhizal fungi. They don't make mushrooms, which ectomycorrhizal fungi usually do. These arbuscular mycorrhizal fungi live inside of the plant root, and they form these structures inside of the plant root cells that you can only see under a microscope called arbuscules. And these are like little itty bitty tiny hyphae that branch out into almost like this little bush inside of the cell. We believe this is where the exchange takes place between the fungus and the plant, where the fungus can provide the plant with particular nutrients. And in case of AMF, we're usually thinking about phosphorus. Now, that's not the only structure AMF make. They also make these other structures inside of the root cell called vesicles. That's their storage structure where they store things like lipids, for example. And then they have hyphae running through the root. So if we look at a piece of the root running through the root, connecting all these different parts, and then they have hyphae out in the soil that are working to absorb nutrients and other key important things from the soil that the plant needs, but mostly phosphorus. They reproduce what we call asexually, and they do that through spores that are mostly produced underground. And you usually need like a microscope to see those. Sometimes you can see the spores by sight with your eyes, but they're almost like the size of little dust particles. It's really difficult to see. So these arbuscular mycorrhizal fungi, about like of all the 90% of plants that associate with mycorrhizal fungi, like 80% of them are with this group. And there's a really strong evolutionary history between arbuscular mycorrhizal fungi and then the plants that they live on. So even though in this relationship, it's thought that the AMF needs the plant, so it cannot get sugar in any other way besides plant material, But the plant necessarily doesn't need the fungus. It only might need it in certain situations. But in prairies, there are some plants that almost can't survive at all without their mycorrhizal partner. This is an example of a picture displaying that. So this is switchgrass or Panicum frugatum and a picture by Gail Wilson. These two grasses are the same age. It's just that this one was grown with its mycorrhizal partner in the pot and this one wasn't. So you can see, even though it might not be completely needed or the plant can still be there, it's basically the plant can't survive without mycorrhizal fungi. And that's common for a lot of grassland and prairie plant species. These mycorrhizal fungi are really important. And we already know some key things about arbuscular mycorrhizal fungi in prairies. So in prairies in prairie plants, for example, that show up mostly in remnant prairies, so those late successional ones that I showed you in the earlier picture that are difficult to get back in a reconstruction, we know that AMF are actually more important to these plants than they tend to be for some of the more weedier species in a reconstruction. And this graph down here is showing that. So each of these dots represents a different plant species. And what Liz Kojal did here is she organized them by successional stage. So you could think of very early as like the weedy plants in a reconstruction, and then the late as like the ones that only show up in the remnant sites. And on the y-axis here, we have mycorrhizal response. So how much better it grows with mycorrhizal fungi from the prairie versus without. The early ones are about a 1. They don't really care so much. They grow kind of the same with or without their mycorrhizal partner. But you'll notice that the plants in the late group grow on average about 7 times larger with mycorrhizal fungi. And some, like this point up here, which Liz says is an allium, it's a native onion plant, grows 25 times larger. Basically, it doesn't grow if it doesn't have its mycorrhizal partner. So we know that for those plants that have trouble showing up in the reconstructions, the AMF is really important to their survival. We know that different AMF species interact differently with different plant species. So it was originally thought that mycorrhizae was mycorrhizae. It didn't matter who it was, the plant would be interacting with it. But we were learning that there's some specialized interactions there where it might actually matter who the mycorrhizal fungi is. And there can be potentially 10 to 20 different mycorrhizal species living on one root system. And we're still not exactly sure what to call a species when it comes to arbuscular mycorrhizal fungi. And we also know from previous work, and I'll talk about this later, that if you take prairie soil, small amounts of soil from a remnant prairie, and you put that with a plug for one of these later successional native plants, that it helps it grow in the reconstruction. So this also suggests that there's something in those remnant sites, something different about their microbial community in that soil than we find in our reconstructions. So I know I've talked about this study a lot with this group, but I think it's important because it also involves Missouri Prairie Foundation prairies, and it kind of gets at the idea of like, do we need to think about the mycorrhizal community when we're thinking about reconstructions? So for my dissertation work, when I was at the University of Missouri in Candy Galen's lab, this is a question that I really wanted to address. We know that in these prairies, adding native soil from a remnant site can help plants grow in a reconstruction. But at the time, we weren't really sure what the differences were in the communities of mycorrhizal fungi in these different places. And genetic sequencing was becoming more popular and was a great way to kind of get at these questions. So one question I asked in my dissertation was, are the mycorrhizal communities, or the number of species and which species are dominant in the mycorrhizal community, different in grasslands with a history of agricultural disturbance? So to ask this question, I actually worked in 2 different habitats. I worked in prairies, but I also spent a lot of time during my dissertation working in glades. So I included those 2. And if you're not familiar with glades, glades are also a grassland, but they're a little bit different than prairies. We find them here in Missouri in the Ozark region on the south and southwestern facing slopes of the hillside. They're really, really rocky. So the soil is really shallow, but a lot of the same plant species exist in both habitats. So I looked at the mycorrhizal community in both prairies and glades. And then I was interested in how land use history affected the mycorrhizal community, which means something very different for glades than it does for prairies. For glades, because they're so rocky, they're so dry, some people call them the deserts of the Midwest, or the deserts of the Ozarks, they don't really experience large amounts of agricultural disturbance. They're hard to get to. It wouldn't be really easy to plow. Their main threat is just absence of fire. So we know Native Americans used fire here to manage their landscape quite a bit. And with the onset of European settlement, differences in cultures caused a lot of these places to fill in with eastern red cedar when the fire was no longer moving through the system. But what a lot of land agencies can do is if they find a glade or a place that they used to think was a glade, they can remove the cedars, return fire, and a lot of times the plant species will slowly start to come back on their own, a lot of times without any seeding. So in this experiment that I did for land use history for glades, I considered a site a remnant if it had never been filled in or had been open for 30 years or more. And I considered a site kind of a new restoration if it had only been restored in the last 10 to 15 years. For prairies, land use history means something a little bit different in this experiment that I did. So I considered a site a reconstruction or a restored prairie if it needed some type of seed addition. So these were sites that usually went through some type of crop rotation or went through heavy grazing in a way that it changed the plant community enough where seed additions had to be added to restore the prairie. Remnants would be places that seed additions were never added. So these would be sites that were either that were never used post-European settlement or were used as hay fields. So the soil was actually never disturbed in any great deal. And then I also looked at this across soil substrate or pH, because we know that soil pH can affect the interactions between plants and mycorrhizal fungi. And both glades and prairies are found on different soil substrates and specifically geological substrates. So I looked at the difference between sites on sandstone, geological substrate, and sites on more, which would be acidic, and then sites on dolomite or limestone, which would be considered calcareous. And I did this for places both in the Ozarks and in the Osage Plains down in the southern part of Missouri. Okay, so I collected samples in many different ways, but what I'm going to focus on today for my results are the community samples, which are samples that I collected roots from plants. I didn't worry about who the plant was. I did collect species-specific samples, but the results were really similar. So I'm just going to talk about one of those today. So what I do in this is I collect samples, I extract DNA from the soil or from the roots. In this case, in this experiment, it was from the roots specifically, all of the DNA. And then I use special molecular techniques to just focus on the mycorrhizal fungal sequences of DNA. And then we use a machine called a sequencer. In this case, we're using next generation sequencing platform to figure out or to get all of these sequences. And then we use those sequences kind of like you use barcodes at the grocery store. With using large computers, we take all of that data and use the sequences to tell us something about how many species are present. Now, we're not exactly sure what a species is, so we're using genetics to try to tell the difference between different types of mycorrhizal fungi or arbuscular mycorrhizal fungi. And so for the community for this, what we look at is we use these graphs that kind of tell us something about community composition. So they take into account both diversity, but then also which species are most dominant in a community. So in these graphs, each point is going to represent the mycorrhizal community at a specific site. If 2 points are closer together, that means the mycorrhizal communities at those sites were much more similar. If a point is further away, it means that they're different. And then I'm going to highlight them by certain colors that tell us which of the groups they're in: habitat type, soil substrate, or land use history. So in this graph, I'm going to show you glades are going to be circles, prairies are going to be squares, and then the pinks and oranges are acidic, the blues are calcareous, a lighter color means degraded or restored, and then a darker color means remnant. So the first thing I want to show you is in this graph, you'll notice that sites that are glades, for glades, soil substrate matters. And there was a huge difference in the soil pH between acidic glades and calcareous glades. So for glades, how long it had been restored didn't seem to be a factor in determining the soil community composition for mycorrhizal fungi. It was more about the soil pH. But for prairies, we got a different story. They tended to separate not by soil substrate. And in fact, we found that in these prairies, pH wasn't as different between acidic and calcareous geological substrates, but it was more about the land use history, sites that had had some form of heavy grazing or some type of large agricultural disturbance like monoculture, corn, soybean rotations, had a different mycorrhizal community than those in remnant prairies. And it wasn't necessarily that the diversity was different, it was that they had different individuals that were dominant in these places. So they had different mycorrhizal communities, different individuals, and then different groups that were the dominant member of the community. And we could even see some separation between sites that had a history of row cropping and then sites that had a history of no row cropping. And that's usually what we consider for grazing. I'm gonna go ahead and skip this slide. Okay, so in glades, AMF community composition varied across soil substrate for prairies, but it was more about land use history. So this suggests that maybe because the community composition of mycorrhizal fungi is different in these places, maybe the plants that we're interested in restoring or getting back, maybe one of the reasons it's difficult to get them there is because the mycorrhizal community that would usually support them is either not present or the certain species that they interact with are really, really rare. So then this asks, well, if AMF communities are different in reconstructed prairies than remnants, how can we restore these mycorrhizal communities that support our plant communities? So here, if Felicia doesn't mind, I know we only have like a couple minutes left. Do you mind running that poll really quick?
Yeah, sure. I just launched it.
Oh, okay. So if you guys could fill out, it says choose your own adventure. Would you like to hear about inoculation first or changing the soil environment first? Can they vote? They might not be able to.
Yeah, they're voting. It's coming in.
Oh, it's coming in. Okay. How many more minutes do I have left, Felicia?
Oh, you've got probably, let's see, like 8.
8? Okay, great. That's good.
All right. It looks like 76% have answered, and they want to see inoculation with prairie mycorrhizal fungi.
Okay. Okay. Thank you. Awesome. So I'll go ahead and go there first. Okay. So is there a way? So let's think about this for a second. So one way we could think about getting these mycorrhizal communities back is by actually taking mycorrhizal fungi from a remnant prairie and moving it into these reconstructions. So, and we know from previous research that that is helpful, and I'll show a graph for that in a second. So is there a way to sustainably reproduce and utilize native prairie mycorrhizal fungi from prairie remnants? So first, I want to say that we know from the literature, and this is a study by Middleton and Beaver in 2012, but multiple experiments have showed this, that especially for those later successional plants, that taking small amounts of soil from a prairie remnant and then taking it and moving it with the prairie plant into the reconstruction can increase survival and sometimes size of the plant. And this is a graph showing the difference in survival in Liatris spicata between prairie when it's planted with prairie soil, as opposed to when it's planted with soil that's been sterilized, so it does not have those prairie microbes present. So we know that like if we take the actual remnant soil, that makes a difference for these plants. But you might be asking, but we can't just go up and dig up the whole prairie. Is there a way that we can almost like make inocula that includes the mycorrhizal fungi that's important for restoration? And there is so the lab I did my postdoc with at the University of Kansas, they've been working on this for quite a long time, this mycorrhizal inoculus from prairies. Jim Beaver and then also his student Liz Kozel has spent a lot of time working on this. And what they do is they take a small amount of soil from a prairie remnant, they plant it in a pot in a greenhouse with a prairie plant, and then they let it grow. And as that plant grows, so does the mycorrhizal fungi in the soil. Then they can take that, look at the spores, and figure out which species are there, or separate it by spores, because we can use spores under a microscope as an idea of different potential types of mycorrhizal fungi, isolate a certain spore, and then grow up species specific, or as close to what we can call species specific inocula. And if they keep doing this over and over again, they basically take a small amount of soil with mycorrhizal fungi and turn it into quite a large amount of soil that has mycorrhizal fungi in it. So that's how they produce AMF inocula in the greenhouse. And I worked on a project with them when I was there with my postdoc that also showed that, and this is some work that Liz Colgill has in review right now that I'm also on this publication. And what she showed was that if you compare AMF fungi inoculated plots to AMF whole soil inoculated plots to non-inoculated plots, and you look at the plant community, so this is forbs versus grasses versus legumes versus plants that died, actually the inocula and the whole soil both do pretty similar. And in fact, this is another graph looking at the plant survival relative to controls. So how much better a plant grew if it was inoculated with AMF fungi only versus the control and whole soil. And you can see that in AMF fungi and whole soil, they both do better. Whole soil did a little bit better in this case, but they weren't significantly different from each other, which suggests that maybe we could just add prairie mycorrhizal fungi and it could do the trick. So the positives of that is we could isolate and amplify mycorrhizal fungi specifically without digging up an entire prairie. And Liz Colgill is working on that specifically right now for her research. The negatives are, for example, we know that when we make these inocula in the greenhouse, they're much less diverse. So the whole soil inocula, if we look at all the mycorrhizal species, there's way more in a whole soil inocula genetically than we find in just the AMF fungi inocula that we produced in the greenhouse. One of the reasons is because not all of these different species of mycorrhizal fungi do well in greenhouses. And in fact, I always thought that they needed a plant and soil to do it. But Liz said that some of the commercial varieties of mycorrhizal fungi, they can grow it in a petri dish just on a little piece of root. So how well they grow in that, and that's not a prairie mycorrhizal fungi, by the way, that's more of a commercial inocula that we usually don't find as often in prairies. So there is some differences in the diversity, but some of Liz's other work shows that maybe there's certain mycorrhizal species that are most important to these late successional plants. So if we can find those particular ones, we might be able to make this work. It also asks, like, how much inocula do we need? So if we want to do this, how can we do it at a large scale? That's still a really difficult question to answer. Liz has been working on that more lately. So she put out a paper in 2020 showing that if you increase the density or how much inocula you have, it does increase native abundance and diversity in your plots. So this suggests that more is better, but then how do we get more is the other question. So that's kind of it for inocula, and I'll answer questions about that more if you would like. And then in terms of the environment, I just want to say we don't actually know a ton about this. Sarah Bear next week might be able to speak about this at a much greater length than I could. This is definitely something she could potentially talk about. But one thing that I've been thinking about is we know, for example, in these reconstructions that the soil nutrient levels are really different. Is there a way to potentially bring the phosphorus levels down? It would maybe haying these sites over and over again, removing biomass. Could that change the soil community or the soil environment in a way that would promote the native mycorrhizal fungi that are important for those prairie plants, or utilizing different preparation techniques? So I'm currently working on a project with the Department of Conservation looking at adding cover crop agriculture prior to a restoration to see if that changes the soil community in any way. So I think I'm going to leave it at that and take any questions that people have. Does that sound good, Felicia?
Yeah, sure. I'm going to unmute Carol here.
Okay.
Just a second. I'm trying to.
And while you do that, I'll just say, I'll have a couple conclusion slides I'll just bring up really quick.
Sure, that's fine.
Okay, yeah, I just wanted to kind of say my general conclusions are, I just wanted to kind of stay at the end, is that arbuscular mycorrhizal fungi and soil microbial communities in general need to be considered in prairie reconstructions. I think a large body of work supports that. And not just any fungus works. So we know from previous work that its local species might be important, specifically ones that have evolved with prairie plant species in particular. So who the partner is in terms of the mycorrhizal fungi might be really important in restoration of prairies. Adding native mycorrhizal fungi to prairie reconstructions will most likely improve its establishment, but the best methods for large scale is still being explored and figured out. And then mycorrhizal fungi, I just want to add that I think mycorrhizal fungi make the world go round. The University of Kansas, Liz made us a shirt that said that. It was pretty funny. But there's lots of other parts to consider, and especially in the soil community. And in fact, like I said, I think Sarah Bear could talk to that too, about that too. So I study specifically mycorrhizal fungi, but fungal pathogens also are really important to maintaining prairie diversity. So it's something to consider too. So I'll leave this up while we take questions. It's just my thank you page.
Sure. Alice, can you hear me?
Yeah.
This is Carol David. I'm the Executive Director of the Missouri Prairie Foundation. And thank you very much, Alice. That was wonderful. And we do have some questions. I'll just dive into these. A question from Meredith. Asking, will we record? And yes, we did record this. And Alice's presentation will be available at the Prairie Foundation YouTube channel. And Felicia, tomorrow, she'll send an email to all the participants with that link and some other information. A question that Felicia has is, how many prairies did you visit to do this? Your most of your.
So I know I visited, I believe it was 40 sites, but only about half of those were prairies. So I think it was around 20. And some of those locations were like, I would have a restoration or a reconstruction and a remnant at the same location. So if I go back to, let's see, I'll find my map. This map shows all of the sites, and the prairies are the squares. So you can see I'm kind of focused in here, and then all of this, these are the prairies on the side. You can see a couple of acidic glades mixed in there. But some of these sites were the darker color and the lighter color on top of each other. That's a site that had a reconstruction right next to a remnant. So I actually sampled both of those. So I think it was around 20, and I can't remember the exact number for that. But many of those were Missouri Prairie Foundation managed prairies.
And we are very proud that you were able to do your research on some
yeah.
And it underscores how important these original remnants are. And some of these remnants might be rather small, but because they're undisturbed, they are gold mines of information about soil microbial life.
Yeah, definitely.
We do have some other questions here. Could you develop a system to pull small soil plugs from a native prairie, then plant them in a matrix for prairie restoration?
So it's almost like it would be like I'm assuming this is someone's thinking maybe like a large scale, more industrial, quicker way to do this. That's a really good question. In terms of plugs, from what I have done and what my colleagues have done, we usually take small amounts of soil and from different locations so that we're not disturbing the prairie too heavily, and then mix that soil and use it in the greenhouse or wherever we're growing our plugs, and just add a small amount in there. That's a good question. I don't know. I'm sure you could. And I bet there's somebody out there who's tried. I've never tried. I have heard of something called seed balling, where they almost take parts the seed when they mix the seed for a reconstruction. They also kind of mix it with some of the upper layer of the soil and create almost these seed ball things that they release out into the site. That might be similar to what you're thinking of. So that could be an option. Yeah. And I don't know if anybody's tried that specifically.
One thing I did want to add is I would exercise extreme caution in doing that, just because anytime there is any kind of soil disturbance, that's an entry point for invasive plants to take hold, because there can be seeds of Sericea lespedeza and other invasive plants that are just waiting for the opportune moment. Not to say that some bare soil isn't natural, and it is, and it can be beneficial for many insects and things, but I would exercise caution on doing something large scale like that.
And that's a good point, Carol. I want to add to that. Like, so when I take my samples, for example, in glades, I would always try to take really small plate, samples. And when I go back, I would make sure, can I see where I've been before? And one time I came back to a site and I think somebody must have been there looking for echinacea bulbs. I'm not sure, but I came back to the site and I was like, did I do this? Because rocks were moved and I could see huge patches of brown. And then I realized that somebody had been by recently looking for echinacea bulbs at this site. And when I revisited that site for a couple of years, those locations where that huge disturbance occurred never quite looked the same.
Yeah, that's right. And glades are very sensitive to that kind of disturbance. We've got some more questions here. Does AMF help prevent problems from pathogens? Oh,
that's a good question. I know people have looked into that. I haven't looked into it in a while, and I don't have very specific, I can't recite any specific research for you. I have seen some work that shows kind of it can go either way. So for example, if you think about mycorrhizal fungi, they have a cost to the plant. So the plant does have to provide resources to the mycorrhizal fungi. So depending on the situation, sometimes it might make it more difficult for the plant to handle a pathogen. But there are situations, for example, with like nematodes and other things where they find that when plants have a mycorrhizal partner, that in fact they are better at handling pathogen loads, and it could be because of the extra resources that the mycorrhizal fungi is also providing. So I think it can go both ways, and it probably depends on the environment. But there is a lot of work on that, and I don't know all the specifics of it. It's a good question.
Thanks. Another question: were any of your prairie sites wet prairies, and if so, did that impact your results?
No, most of my sites were not. Yes, it probably would. So there's a couple ways that could impact it. One is that sometimes that changes the way these mycorrhizal fungi might disperse. And it depends on how wet. So we think that potentially in really drought-prone conditions that mycorrhizal fungi might help with water movement and take up of water for some plants. And holding on to water for some plants. So in those wet places, that might not be as important. But however, at the same time, fungi in general are really limited by moisture. And in fact, like if they don't have moisture, it can be difficult for them, and they can what we call desiccate or dry up. So it definitely and would most likely change the results. It could have been a completely different community of mycorrhizal fungi in some ways. So I did not look at any of those, and it would be really interesting to do that.
Christine has a question. A friend and I have some property next to the Mississippi River. We are starting a fruit and veggie farm and have used some commercial vegetable mycorrhizae for our veggies. Could that possibly help or hinder native plants that we plant to attract pollinators?
Good question. So in terms of attracting pollinators, that is just starting to be looked at. So like how the below ground mutualisms affect the above ground mutualisms. The only work that I know of for sure that's been published that I can think of, though I'm sure I'm missing a lot of things, was it showed that it changed the interaction for nectar robbers. So actually ants up in the alpine, their interaction with flowers changed based on mycorrhizal inocula present on those plants. The commercial inocula Carol, is that person asking for their veggies particular, or are they thinking about using that same inocula for their prairie plants?
Christine doesn't specify. Maybe if she's still there, she can send another follow-up.
Okay.
But I think it sounds like she's using the mycorrhizae for the veggies, but the guest is maybe wondering if it might, affect native plants nearby, is what I'm.
Oh, I see.
Guessing.
Okay.
I'm guessing.
Yeah, so it depends on the commercial inocula. There's some research that suggests that for some commercial inocula, so to really produce these at really, really large scales, oftentimes you need to use a species of mycorrhizal fungi that reproduces really easily in the greenhouse, or like I was saying, Liz was talking about in a almost like a petri dish. And it just happens that a lot of those that are really good at reproducing in those environments also tend to be less beneficial to native plants. That's not always the case, but a lot of times it is. So in terms of commercial inocula, there's lots of different kinds. And in fact, there are commercial inocula available for that are developed from prairies specifically. So it depends on the inocula. And in fact, some of the more commercially produced large-scale inocula, some research suggests that a lot of the propagules or the spores, by the time they get to the consumer, are dead. So it could affect the plants around it. And in fact, I did some research in glades that showed if I inoculated a particular spot, seedlings half a meter away could be impacted by that inoculation. And some research in prairies has gone as far as 2 meters. So in regards to the surrounding community, it could definitely affect it as well. But in terms of how, it might depend on the specific type of commercial inocula that you're using.
Kind of a follow-up question here from Dan. Is there any concern that these commercial products could become, quote, invasive?
Yes, it's so hard, and we have to be so careful when we start moving microbes around, especially because for mycorrhizal fungi, one of the reasons we think they might not make it, or why the community is different than a remnant. And some of these sites I worked in, the remnant and the reconstruction were just like on the other side of a road or right up against each other, is that it takes a really large soil disturbance to move them, or they just move really slowly through their hyphae or their spores, which are below ground. So maybe, I'm not exactly sure how they move, maybe animal disturbance, maybe like small insects like columbella could move them. And so when we start moving them from place to place, we do have to be very careful. And in fact, there are some mycorrhizal fungi that are really good at reproducing and actually decrease growth of some plants as opposed to increase it. So they almost act like a pathogen. So we do have to be careful. It's a great question.
Another question, what plant species or taxonomic groups do not associate with AMF?
That's a good question. So the group that I think about most is brassica. So mustards. They do not. Good question.
Thank you.
I think there are other ones, but that's the main one that I think of. Like orchids, for example, they're mycorrhizal, but in a really different way. It's a totally different group of mycorrhizal fungi, and the interaction is very different. It's almost the fungus is kind of providing the carbon, which is a different scenario. And then there's lots of plants that are ectomycorrhizal partners. So lots of, like, for example, oaks and hickories and woodlands, pine trees. In prairies, though, probably mostly brassica would be what we would consider a non-mycorrhizal species.
Great, thanks. Boy, we've got more questions. People they're very interested. Matt is asking, is the fescue endophyte considered AMF?
I'm not sure if I know what Matt's talking about.
Okay, so tall fescue grass, there's a fungal endophyte. There's a fungus that lives within the plant. I believe in the plant cells. So I don't know. Definitely in the above ground parts. I don't know if it lives in the roots.
Okay. Okay, so if it's above ground, it's probably not, it's not mycorrhizal fungi, but that doesn't mean it's not a mutualist. There are tons of other types of fungi that live both in the above ground and below ground parts that do all different types of stuff in plants. So I'm not exactly sure what this particular one is. I'd have to look into that, but so I don't know. I'm sure it's not mycorrhizal fungi if it's in the above ground tissue.
We'll put in, I'll see if Felicia can put this into the email that goes out tomorrow, but we had a whole issue of the Missouri Prairie Journal devoted to fescue. And there's a lot of here about the fungal endophyte, so people can learn more there. A related question, is it true that fescue thrives in bacterial soil and actually depletes mycorrhizal fungi? Would a specific cover crop that would suppress fescue growth benefit mycorrhizal fungi?
That's a great question. So fescue, we think most likely does, it's not as what we call mycorrhizal as some of our later successional prairie plants. So it doesn't rely as heavily on mycorrhizal fungi as some of our later successional plants. It does use mycorrhizal fungi, but in a very different way. So yes, it most likely suppresses that community when it's in monoculture. And there's been some work looking at how non-native monocultures and then restorations afterwards impact the mycorrhizal community. And they can have large effects depending on what plant species is there. And then in terms of the cover crops, that's a really good question. So I mentioned that I was working with the Missouri Department of Conservation on that. And my collaborator, Elizabeth Middleton, who's now in Indiana, after my work in the Missouri Prairie Foundation prairies on mycorrhizal communities, we thought, well, maybe if some of these important prairie mycorrhizal fungi are actually present in these reconstructions, they're just really, really rare, maybe changing something about how we do a reconstruction could make it so they could do better when the prairie planting happens. And so we were trying to explore cover crops as an option, because we know in agriculture that that's used a lot to improve soil health. But soil health in agriculture is very different than soil health in a prairie. For example, the fungal to bacteria ratio is much lower in agriculture, just like you mentioned in fescue dominated places. So we think that some cover crops could maybe help. The only thing that is hard there is those cover crops tend, not always, but tend to be annuals, which annuals tend to invest less in a mycorrhizal community because they're more focused on seed production and reproduction quickly, rather than putting in a lot of energy into their below ground carbon sources. So maybe. And so that's what we were trying to explore there. Unfortunately, our site made it really difficult to get cover crops established, but we are going to look at how the metrics used in soil health and agriculture, so things like biomass and other soil health metrics like the Haney test for nutrients, how those measurements correlate with mycorrhizal communities in both prairies and agricultural locations. So it's a great question. I don't think we quite know. I think maybe. Mm-hmm.
Thanks. Some more questions here. Sam asks, do remnant sites show a higher resilience to invasion from non-native plant species due to AMF?
Maybe. Maybe. So I think from what I've learned and from the literature in general, it's probably both mycorrhizae, but also pathogens. There's a lot going on. It can also be competition with those plants that are already established. And a lot of plant ecologists work on competition. I always think about things in terms of mycorrhizal fungi and plant microbes. But we know, for example, that what happens in these places and why pathogens are really important in prairies too, is they can keep things that would take over from taking over, because as that plant reproduces, so does its specific pathogen in the soil. So it almost like keeps it in check. So yeah, mycorrhizal fungi might play a role because it gives some of those plants that use that mycorrhizal fungi an advantage that they wouldn't have if that wasn't available to them, specifically those ones they've evolved with for a really long time, like the ones in the prairies. But it also could have something to do with pathogens that are present there as well. And both of those things, as well as competition with the plants that are already present. So that's probably a really that's a great ecological question that a lot of people are trying to figure out.
Hey, Bev asks, is it difficult to extract DNA from soil?
Oh yeah, no, luckily they've come up with some great kits to do that. Now you do need a really sterile environment and the right kit, which can be pretty expensive. And you do need some very specific tools. So for example, we have a machine in the lab that we use to shake up these tubes with the roots in them, or the soil if we're using soil, to break the cells apart so that we can actually get to the DNA inside of the nucleus. So you do need some specialized things, but you can also just use a different type of machine to shake it really hard, and you could probably get a similar scenario. So if you have a lab, no, it's actually not too hard at all, because it's kind of like following a recipe. There's lots of kits that you can use that tell you exactly what to put in there at a specific time. And the different steps, the different steps do is they break apart the cells, break apart the nucleus, and then isolate and remove everything but the DNA. And so that's what the chemicals that you use in the process does. And there's lots of different kits you can use. So if you have the lab space, it's not difficult at all. It's kind of like, cooking dinner, following a recipe.
Wonderful. Another question. Has there been any data collected from other regions? How similar would the fungal diversity in Missouri be compared to a prairie in Minnesota?
Yeah, great question. So there was a a student who, for their dissertation, asked a similar question to what I did in Missouri, but asked it across a couple different states in restorations and remnant prairies. And now I can't remember in terms of how diverse things were over in different areas. So for example, they went all the way down to Oklahoma, which the soil down there is so different. But what they did find, even across all of those places, was a really similar result to what I found, was that reconstructions were different than remnant prairies. And in fact, they found that diversity increased with precipitation, but that was only true for remnants. Like in reconstructions, it just that scenario just kind of went away. It was like all of the disturbance totally changed how diversity or community composition changed across the precipitation gradient. So in terms of the differences between reconstructions and remnants, yes, there is differences, but also pH plays a role in diversity. So the pH of soil, for example, in Oklahoma prairie or in a Minnesota prairie might also alter that a little bit. And that's a good question. I don't know about diversity specifically. And diversity for this genetic work is really hard to get at. We try to use this information to tell us about diversity, but because of the way we collect the data, it's a difficult thing to assess. It's much easier to assess composition. But that's a good question.
Another question. When did you take into account the population of invasive species? If so, did certain invasive species show any impact in mycorrhizal population?
Good question. I have not actually looked at invasive species in my research specifically. However, when I was at the University of Kansas, we set up reconstructions, and I think I have a slide somewhere of this. I worked on a project where they set up reconstructions at multiple locations in different states. Moving way ahead, ignore, don't get sick watching this. Here we go. So we set up reconstructions in multiple locations in different states. And some of these reconstructions were areas that had been planted with fescue prior to restoration. And then down in Oklahoma, they also have this plant called Old World Bluestem, which is a non-native, but it's actually really mycorrhizal. And what we found was when we were looking at our plots and we were trying to determine how inoculation moved from the point of inoculation, we found that in places that had been invaded by Old World Bluestem, that we saw a lot of encroachment from the edge of our plots where the Old World Bluestem still was in terms of mycorrhizal fungi. So different invasive species change it in different ways. And Gail Wilson is a researcher who's looked at that specifically in Kansas and Oklahoma. So if you're interested in that, I would look into her work, but that's a good question. And it definitely changes it.
Great, thanks. Ariel has a question. What is a good resource to learn which plants belong to which successional stage? And would it make sense in a very disturbed habitat to start with early successional plants before adding in the later ones along with AMF inocula?
Yeah, that's a good question. There has been some work on trying to figure out if it's actually worth even putting in the seed of some of these later successional plants early in the restoration process. I don't know, Carol, you might actually have a better idea of a good resource for that than I would.
Well,
no?
Well, I have an answer. It might not be exactly the best thing, but I don't know of a resource that will look at early successional versus late successional, but you can look at a list of plants and their coefficient of conservatism, which is I guess you'd say a value system or sort of a system for assessing the fidelity of a certain plant species with a remnant ecosystem. So for example, ragweed might have a number of 1, whereas grass pink orchid might has a number of maybe 8 or 9. And so the higher the number, the higher fidelity to a specific remnant community. In other words, the more dependent it is on a stable, intact remnant community. And of course, some of the, some species are really hard to propagate, like orchids, but I would also say that we have an article in the current issue of the Missouri Prairie Journal about 3 reconstruction projects that we've been undertaking right now. And a lot of times what we do is we might add, do a seeding mix of a variety of species, sort of the matrix species that aren't, super low in conservatism, but not super high. But then we might add plugs of species that are higher, that are more conservative because we have a higher success rate. They're just more expensive to, gather seed from and that sort of thing. So I don't know if that answers that part. And I will look for that coefficient of conservatism list. And so Felicia can share that tomorrow in the email.
Yeah, and I think, Carol, what you said too, I think if I remember right, that graph that I showed, I'm pretty sure Liz Kojal used that conservatism number to get those categories. So that makes sense.
Okay, great. It's 5 o'clock. Do you have time, Alice, for just a couple more questions?
I can take maybe 2 more.
Okay, and unfortunately, I have to sign off now, but Felicia's going to address those 2 questions. And thank you so much, Alice. And Felicia will finish up here.
Okay, thank you so much.
All right. Natalie asks, have you done any study to see if mycorrhizal fungi have as much effect on forest growth as it does on the prairie growth?
I have not worked in the forest, but some people have, and it's just like in the prairie where it depends on the species. In fact, when we're thinking about Midwestern forests or woodlands, it depends really if you're talking about the understory or the trees, because in oak hickory woodlands, they would be associating with ectomycorrhizal fungi, which is a completely different group. Those are ones that sometimes a lot of those might be able to live without a plant as well. They can switch back and forth, and they produce mushrooms usually. And so, yeah, in forests, they can make a big difference depending on the species. And I haven't specifically looked at it, but a lot of people have.
All right. Okay, last question. And there's a couple more, and I'm just going to copy them down and we can address them later.
Okay.
This last one here, is there any research regarding prairie management tools like fire, grazing, haying, etc. affect AMF?
Yeah. Oh, that's such a good question. I should have looked back at the literature before I came to this talk. Yes. All of those have been looked at in various regards and in various places around the world. So for example, I know that grazing has been looked at heavily in China and in grasslands in China. It seems to depend on the intensity. So for example, there's a researcher named Nancy Johnson, and she worked on looking at how the intensity of fire might impact mycorrhizal fungi. And what she found is a longer study, but what she found was in places that had been scorched really heavily, if the fire was really intense, it would potentially, it seemed like mycorrhizal fungi were gone in those locations. But at the same time, we think about prairies, fire, I mean, all of these species, including the microbes, are adapted to fire. So for fire, it seems like intensity might play a role. I tried looking at how long it had been since a fire for the mycorrhizal community composition in the prairies that I studied, and I did not see any strong, well, I saw a relationship, but it wasn't as strong, for example, as the difference between reconstructions and remnants. So that won over fire and like the time since last fire. But I have seen some studies that show for both grazing and fire, intensity is really important. Who was the grazer? How much? How strong was the fire? How hot did it burn? And is that ecosystem adapted to fire? So all of those things play a role there. That's a good question.
Awesome. Well, thank you, Alice, so much for your time. This was an excellent presentation, as always. And we had great questions from the crowd. Tomorrow, I will send out a follow-up email in the afternoon with a recording and some references to some other material.
Okay, great. Thank you.
Thanks a lot, everyone.